Good morning. Is it visible? The signal transduction slide.
Yes, ma'am.
Yeah. So let's start. Previous day we were telling that the signals, how cells receive signal. So it is the mode of action by which the cells enact in a synchronized manner how they just get any signal, how they pass on the signals to the next cell, as well as intracellular signaling cascades are on. This is our topic to be started today. If you just go through this picture, the cell, the ash colored bordered portions is the cell. This one the intracellular portions of the cell, and these are the outer cellular portions of the cell. And the cell, it expresses the receptor. These green color cup like things are the receptors, and the red one is the signaling molecule. It may be a chemokine, cytokine, or suppose there are antigens which have been degraded into the antigen presenting and processing cell, like the phagocytes, like the macrophages, like the monocytes, dendritic cells. They can engulf the foreign pathogen, they can degrade the foreign pathogen inside them to make the larger proteins into a cleavable matter of small fragments of peptide. And these peptides, when they displace over the cells, they are recognized by the different populations of the cell, which are responsible to give rise to the immunological response. Now, what happens if the signal, it may be a chemokine, cytokine, or a pathogenic component, whenever it sits over the receptor on the cell, then the signaling cascade becomes on, and the intracellular signaling molecule B, it is molecule A, it is the extracellular signal molecule, and this one in the intracellular signaling molecule. And by as the baton is passed from one person to another in a relay race for signaling transduction, signal transduction methodology, these sort of passing the signal molecules on from one side to another side, it occurs like this. Now, the gland, previous day we discussed about this gland. Can you remember? And somebody was asking me about the exocrine gland. Can you remember this one? The 4 ways of signaling in multicellular animals: endocrine, paracrine, and neurocrine, paracrine, contact dependent or juxtacrine methodology of signaling cascades on. Can you remember this one we were discussing? I would just recapitulate how the methodology of signaling molecules secretion and their passage from one place to another in vivo system. In vivo means inside the body system. How does it occur? Now, if you just closely look into this picture, the left hand one is the endocrine system and the right hand one is the exocrine system. Endocrine system, here it involves the blood capillaries, means the hormones are the example of endocrinally passed chemical which acts in a different manner through the blood or lymphatic capillaries. And what happens, all the chemicals which are secreted from the endocrine glands, they act like this. Just have a look. The endocrine glands, they secrete the signaling molecule and they take this one through the bloodstream from a place to a distant place, where in the target cell they just act on. Now, uh, depending upon their secretion methodology and the delivery methodology, if you look this one, the endocrine glands look like this when they just secrete. Can you see properly? This one, they are secreting some chemicals, and these chemicals are getting outside the cell, and this is the capillaries, blood capillaries. And whenever these cells are captured by the capillaries, these blood capillaries, the blood vessels, they take these chemicals from one side, from the origin of their development to the target cells. So a distant way they act on. And the exocrine glands, they just act like this. It is a exocrine gland, and the chemicals produced by the gland is secreted directly just outside the cell. It is a cell, epithelial cell, endothelial cell, whatever cell it is there. So whatever it is producing, the signaling molecules, the chemicals, they are secreted, and just outside the cells they are released. There is no way for carrying them to a distant place. But in case of endocrine system, they are taken distantly, where these chemical has to act on a target cell. Now, the glands which are exocrine in nature, they are of these variety: merocrine, apocrine and holocrine. I will be discussing later about the names and why they are named like this. The apocrine glands, they are secreting the materials in a vesicular encapsulated manner, and holocrine, the whole cell is burst. Can you see the whole cell after developing some secretory material, secretory chemicals, chemokines, the whole cell bursts and the chemical is released nearby, and by destruction of the whole cell, killing of the whole cell, these holocrine glands act. And the merocrine glands, they act like this: a small chemical just released outside the cell. Apocrine one, they are releasing vesicle-like structures encapsulating that desired material, desired chemical. Can you understand the basic difference of the methodology? And if you look in the right hand panel, the autocrine signaling system means, auto means self, the cell which is secreting some molecule itself is enacting under the influence of the same material. So the chemical is secreted and that chemical is acting over the same cell. So it is the autocrine manner of cell signaling, chemical signaling procedure. The target and origin cell are the same one. In signaling across the gap junctions. So where two cells are sitting nearby, but there are some gaps, like the nerve cell in the synapse. Have you heard about the neurons or the nerve cells? Have you heard?
Yes, ma'am. Yeah.
Who didn't have biology in the class 2, I am just telling for them that neurons or the nerve cells have their axons, axons. These axons are very distantly elongated portions of the neuron cells, and these axons ends into the synaptic juncture, where two synapses comes together, but they are not attached directly. So in that juncture, the signaling molecules, because from brain at to the site of your target, means suppose you have a scratch on your hand, and the signaling of chemokine release or the chemoattractant release to fetch different numbers of cells, different populations of the cell in higher concentration, it needs the signaling from brain. So how does the signal passes? The brain have the neurons and the passing of the signals just go from one neuron to another by overcoming the juncture gap. So this juncture gap, it is the site where the signaling cell and the target cells, they meet, but in a gap they have to take the signal molecule, and their passage is meant through a special characteristic features. And in case of paracrine, the signaling molecules, they are released directly, and in the nearby cells, they are the target cells, they absorb over there. So the exocrine system, they act directly, releasing some chemokines from our origin cell to the target cell. And what happens, the signaling to a distance, carrying the signaling molecule to a distant cell, to a target distant cell, it is meant through bloodstream, and indirectly they are passing this signaling molecule to a distant place, and this system is called the endocrine system. Now, there are basic difference in endocrine glands and exocrine glands and endocrine glands. Exocrine glands, they maintain contact with the body surface by a duct. They have a duct. How does this look like? It is a exocrine gland, and they have this duct-like structure, appendages, where inside the gland the chemical is synthesized. They are taken outside through the duct and released just outside the cell. And the endocrine glands, they look like this. This is the glandular structure which is traversed by the vesicular system, means the vascularization. Vesicular means vascularization is there. Vascularization means the blood capillaries traverse in this gland, and by the exchange of the chemical from the gland to the blood capillary, it happens inside only, and the blood capillaries takes the chemokines to the distant place. Now, the difference is the exocrine gland have ducts, endocrine don't have. The exocrine glands carry substance to the membrane surface, and the endocrine glands release hormone into the surrounding tissue fluid directly and picked up by blood capillaries. So these cells, they secrete, mainly hormones are taken distantly to enact over the target cell which are distantly located. And here there is no blood capillary traversion, and directly the duct is carrying this one just outside the cell. The example of exocrine glands are tear glands, sweat glands, and mammary glands. Tear, from the cells of the eye, they are just released and you just have a touch on it. Sweat, the epithelial membranous cells, they secrete sweats and you can touch directly. But the endocrine glands, they do not have contact with the body surface or cavities which lead to the outside, have no duct, secrete hormones directly into the blood. Hormones are the example of endocrine glandular secretion, and the sweat, sebum, tears, milk, these are the examples of the exocrine gland secretion. Now, the mode of secretion, if you just want to differentiate the glands, the merocrine, the merocrine glands, they don't have no part of the cell is lost with secretion. Means the cell becomes intact, remains intact after the secretion. The salivary gland. After secretion of saliva, no gland is destroyed. Apocrine gland, the top of the cell is lost, like they are forming a vesicle or a encapsulated portions. To form vesicles or the encapsulation, the top of the cell is lost with the secretion. The example is mammary gland. So the apocrine secretion does the mode of action in this manner, and the holocrine glands, the whole cell detaches with the secretion, that is the sebaceous glands. So the variety of the glands, they are different and their mode of action is also different. Now going to the slide which were already discussed in our last class. If you remember properly, we will be just going through this one. The endocrine system, where the endocrine cells secretes the hormones and they are taken through the capillary system to a distantly located cells where the receptor of the hormone is expressed. Every cell will not enact upon the hormone. So the cells which are expressing the receptor of the hormone, this green clap-like system is the receptor. It is the endocrine system. Now the paracrine system, here the cell which is secreting the chemokine for the paracrine system, the local mediator is carrying the chemokine to the receptor of the nearby cell. These cells are very nearly located. Just released chemokine will be sitting over this receptor and the target cells will be enacting on this one. Now, if you look at this neuronal neurocrine or paracrine system, that is, the neurons, as their action is very distantly organized, means very long actions, and the neurotransmitter are the chemical which has to be secreted. These neurotransmitter, when they are released in the synapse is the site where the target cell and the neuron cell, there may be two neuron cells passing the signals from one to another, or the last neuron cells releasing the neurotransmitter to the target cell which is nearby located, and same way the receptor is recognizing the neurotransmitter and they are just acting upon these neurotransmitter, and the middle portions, the gap between these two is called the synapse. Contact dependent or juxtacrine is another manner. What is? In contact dependent signaling, a cell surface bound signaling molecule binds to the receptor protein on the adjacent cell. Here the coupling of two cells, it is done through receptor and ligand mediation. So here needs the contact to get it charged, and it is called the contact dependent transmission of cell signaling molecule. Now, each cell responds to a limited set of signals. It is not that lots of signals are released. Chemokines, cytokines, chemoattractants, different interleukins. We will be telling in our immunology chapter what are interleukins. So you just know they are all chemicals, and these chemicals are responsible to create the initiation step for signaling cascade on. Now, which signal is for which cell? The target cell will be identified by the discriminating factor of the brain based on what receptor it is expressing, what intracellular signaling molecule it is expressing, and what effector protein it is expressing. These are the key factor which decides that which receptor binds to which extracellular signaling molecule. But same signaling molecule can bind to different receptor. Means a molecule which are the signaling molecule, they can bind to different type of receptor. But receptor is very much specific. They won't just recognize all the material all the chemicals. Understood the point? Now, if you just look into this picture, the same signaling molecule can induce different responses in different target cell. Means the acetylcholine, it is a signaling molecule. If it interacts over the surface receptor of the heart muscle cell, it decreases rate and force of contraction. Means the contraction and expansion of heart muscle, it is regulated by this chemokine or the chemical called acetylcholine. But the same acetylcholine, it may enact differently. If in case of salivary gland, the receptor recognize the same acetylcholine, it will trigger the salivary gland cells to secrete saliva. They won't contract or expand like heart muscle, but they will release some secretion. The same acetylcholine, when they enact over the cell surface receptor over the skeletal muscle, they will help them to contract. Skeletal muscle expansion contraction is another methodology by which the heart muscle enact. The mode of action is slightly different. So the molecule or the chemical signaling molecule, it is the same. But if the target cell is different, their receptor is also different. So they can enact differently in different manner. The animal cell depends on multiple extracellular signals which can interact with each other. Means, what are the functions the signaling molecule do? You just go through this one. They help in survival of the cell. Means basic cell cycle progression and DNA replication. There are lots of just survival machinery the cell has to perform each and every moment. So they help to survive, to make the cell survive. They make the cell to grow and divide, and they help to make the cell differentiate. We have discussed that the pluripotent stem cells, pluri means different, cells can be generated from the mother stem cell, and upon the activation of different cytokine, chemokine, growth factor, if you change the cocktail of the cytokine milieu environment, then you can regulate, manipulate which target cell has to be developed to meet up your need. So this is called the differentiation. And lastly, the cell can be triggered. The signaling molecule can trigger the killing of the cell also. It is called the programmed cell death or apoptosis. So whenever some cells, their half-life is exhausted, or in feedback mechanism, the functioning of the cell, the body don't need to stop the extra functioning, to conserve the loss of energy. The body always enact in a feedback mechanism. Whenever the function is done, then the mechanism of action, it is stopped. And in case of survival also, the cell follows the same rule. The cells which are needed to be die, it is going to the apoptotic pathway, and the cell faces the programmed cell death. So all are doing its functioning under the influence of signaling molecules. Two classes of extracellular signaling molecules. One is cell surface receptor mediated, another is intracellular receptor mediated. That is, outside the cell, when the receptor is just interacting with the signaling molecule, then the signaling molecule initiates the signaling methodology on, And whenever it is on, the intracellular signaling cascades are also there. And to initiate this signaling, the small hydrophobic signal molecule which are taken without the intervention of the receptor or any intervention over the surface receptor, whenever they are taken inside the cell, the intracellular receptor, they interact with this one and the intracellular signaling cascade gets triggered. Now the cells, how response to a signal can be fast or slow. It should be very immediate or it may take some time to get triggered. You just go into flow chart, to bifurcated flowchart manner. Here it is the extracellular signal and the cell, it is displaying its receptor, and whenever there is immediate response is needed for that management of the situation, the left hand panel, if you just go through the intracellular signaling pathway, it is taking charge, the altered protein is functioning, and the altered cytoplasmic machinery is done, and the altered cell behavior you can see. Altered cell behavior means the cell which are normally intact, no need for any triggering, then it is unaltered cell. And when there is a trigger, this extracellular signal molecule is the trigger, and they may have to release some new secretion or give the secretion to get associated with its nearby cell surface receptor, or it has to be taken distantly located target cell through the blood capillary. Anything it has to perform, the alteration of the cell is needed to secrete something. So alteration of the cell means its triggering is done, and the altered protein functions like this. And if you follow the right hand panel, whenever this cell surface receptor is interacting with the extracellular signaling molecule, the intracellular signaling surface molecules, this is the surface signaling cascade receptor, and here there are intracellular signaling receptor inside the nucleus. DNA, RNA, all are situated inside the nucleus. Can you get the point? So there are nuclear pore, there are gaps in the nuclear envelope. So the extracellular signaling molecule, they traverse inside the nucleus and they act on the DNA RNA material, and the altered protein synthesis happens. Here the altered protein functioning happens. Whatever protein intracellular signaling molecule is there, the protein is itself altered and the cell is giving its altered functioning. But in case as it is altered protein 1, it is giving very fast reaction. But if in genetic level DNA, RNA has to be modified, the alteration in the protein synthesis required, it needs some time, and this time as it is needed, it takes some time to respond, and it is called the slow signaling. It takes minutes to hour, and in fast it takes seconds to minute. Without a minute it is giving the reaction, but sometimes it takes some time where the protein, the altered protein, altered signaling molecule, they has to be transcribed, translated in a genetic level, so it needs some time. Now the steroid hormones have cytosolic or nuclear receptor which act as a transcriptional regulator. That is, the hormones, how the hormones steroid, particularly the steroids, how they act on. It is a cortisol. Cortisol is the signature steroid hormone, and this is the plasma membrane of the cell. This is the inside cell and it is the outside cell space. So the nuclear receptor protein, it is a receptor protein. Inside the nucleus it has to be taken. Can you see this double layered barrier? It is the nuclear envelope. This portion is the intranuclear space, means inside the nucleus it is. Just outside the nucleus it is the cytoplasmic space, and just going outside the plasma membrane it is the extracellular space. Everybody is understanding this cellular organization? Can you have this idea? This is inside nucleus, this is inside nucleus, this is outside nucleus in the cytoplasm, and this portion is outside cell. So the nuclear receptor protein surface protein, it is the surface where the cortisol has to pass by passing the plasma membrane. That inside the cell the nuclear receptor protein is there. These receptor protein just recognize the particular molecule, signaling molecule. Here it is a signaling, we are talking about all signaling molecule. These steroid hormones are also signaling molecule. Now the conformational change activates receptor protein. So the receptor protein, they specifically recognize this molecule, signaling molecule, and their conformational change happens. Whenever this integration is perfectly done, the activated receptor cortisol complex moves into the nucleus. So previously it was not activated. Whenever it is interacting with the signaling molecule, it becomes activated and its conformational change happens. And after changing, after forming a complex, it is carried to inside the nucleus. And the activated receptor cortisol complex binds to the regulatory region of the target gene and activates transcription. Target gene, it has different sites, and the regulatory region where the complex has to sit, they are just recognizing and the complex is sitting over this one. Next step is it triggers the gene, target gene activation, and the transcription happens. As it regulates the transcription, it needs some time, and likewise the steroid hormone, when we apply, it acts quite fastly. Now the intracellular signaling pathway relay the signal from cell surface receptor. What happens? These are the intracellular signaling. This is the cell surface. It expresses the receptor, and over the receptor the extracellular signaling molecule sits. The receptor protein is just touching the lower surface of the plasma membrane and touching the cytoplasmic portions. These are the receptor protein, and inside the cell there are several intracellular signaling molecules. And whenever these signaling molecule they touches with any signaling cascade initiating molecules, they just change morphologically, and the altered structure give them altered function. The metabolic enzyme, they are altered, metabolism triggering, they do the metabolism triggering in an altered manner. The cytoskeletal protein, when they enact with the signaling molecule, the altered cell shape for movement. Cytoskeletal protein means they give the shape and structure of a organ, of a tissue. When it is a unicellular cell, they just help them for movement. On the whole, as the bone gives the conformational structure for a body, the cytoskeletal proteins, they help to maintain the integrity and specific figure, morphology, structure of a cell. So altered shell shape or movement happens if the cytoskeletal protein interact with the signaling molecule. The effector proteins, there are another class of protein that are effector protein. The transcription, what do they do? They change the transcription procedure, they alter, they modulate. So the transcription regulator effector protein, they alter the gene expression, and likewise the cell responses. All the alteration, they help to respond the target cell to enact in a different manner different from its original function and structure. Now the different types of signaling event at the cell surface. The ion channel, there are different type of cell surface receptor, very important. There are lots of time the questions are given that what are the different types of the receptor, how does a receptor function, or a specific receptor functioning like this. So if you just go through the ion channel coupled receptor, so it is a receptor, you can have a look two different type of protein composed receptor molecules, and it is the cytosol, means the cytoplasmic portion inside the cell, and this portion is the outside the cell. Whenever the ions, they are the trigger, and these act as the signaling molecule. Whenever they interact with the ion channel coupled receptor, they enact whenever the ions are interacting with them. Now the ions, when they interact, the signaling molecule, it is not the directly signaling molecule, it has to pass from outside to inside, it has to come inside. So that signaling molecule which are specific for these ion channel coupled receptor, when sits over this one, they alter their morphology, their function, and they just open up. Here it is the closed receptor gate, and it is the open receptor gate. Whenever the receptor gate is open, ions can pass from outside to inside the cell. Next one is enzyme coupled receptor, that is the receptor which enacts, which are triggered when some signaling molecule, they just form a specific type of functioning, and they sits over the receptor, and it is triggered to be enacted. At first, the inactive catalytic domains are looking like this. It is also a signal molecule, receptor molecule, and they look like this when they are inactive form. Whenever the signal molecule form a dimer, in a normal condition, these two molecules are dissociated and they are in the monomeric form. When dimerization happen, they sits over the receptor molecule like this, and the active catalytic domain, the activation is happened by the trigger, and they look like this. They change morphologically, their conformation is changed, and the signaling molecule, they may enact over these, and the third party, the activated associated enzyme, may act over these altered receptor, and they alter function, they alteredly function like this. It is a direct receptor which can have altered catalytic functioning, and it is the indirectly acting receptor molecule. Directly it is acting over the signal molecule, and indirectly an enzyme is attaching with this receptor, and this enzyme enacts. Signaling is done through molecular switches. In molecular level, how does it function? The phosphorylation of protein. Protein phosphorylation, how it happens? When there is signal, molecular signal is there, then the protein kinase, all the S ending, ASC, ending is the enzyme. So protein kinase is an enzyme. whenever signaling molecule enacts over the enzyme, ATP-driven signaling cascade is on. Here, ATP means energy, adenosine triphosphate, energy driven. You have to put some energy, respiratory energy or anything where the ATP is produced, you have to put energy. And from ATP to ADP, when it is formed, means you are putting energy. And ADP to ATP means you are getting energy. Understood the difference? When ATP is produced, means the energy is produced. And when ATP is invested and losing one phosphate from one triphosphate to diphosphate, so one phosphate is released, and this phosphate is just attached with the signaling molecule, with the signaling receptor, and they enact. And whenever the signal is out, means no need for investing your ATP. Unnecessarily body won't invest any energy. So no need for investing energy. Signal is out, then the phosphate is released. And as the phosphate is released, means the activation is off. So to get it off to on, you have to invest one phosphate group, and this phosphate molecule is delivered by adenosine triphosphate. And whenever there is no signal, body gets signal from brain, no need for secreting sweat, it is cool in nature, no need, so no secretion of the cell. That is why they just stop the signaling cascade and the phosphate is released, and no energy is invested to just carry on the circular mannered cell signaling cascade mechanism of action. So likewise, the signaling in a molecular level is happen. Now, glycogen is broken down to respond against adrenaline. Adrenaline is a hormone, it is a stress hormone. When you are going to the examination hall, when you are just passing a lonely night outside your home, any stress condition, you are facing any enmity or any stress, the hormone adrenaline is secreted. What happens when adrenaline is secreted? This red dot is the hormone, and it has secreted from a distantly related cell, and through the capillary or the blood system, the blood vessel carries to the targeted cell. This one is the targeted cell cytoplasm. It is outside the cell, it is inside the cell, and the target cell express its specific receptor. You can see the receptor looks differently, and the activation of the receptor happens only when the adrenaline sits over this one. The activated adenyl cyclase, another enzyme which triggers the different signaling cascades, and it is also energy driven process where ATP is invested, and through cyclic AMP the ATP is just triggering the cyclic AMP signaling cascade on, and inactive PK, it is also one step of signaling cascade molecule, and whenever this cascade is on, the energy driven signaling cascade is on, the inactive phosphorylase kinase, it is another enzyme which is remaining in the cell in an inactive form, and whenever these activation of the signal the cell gets, the phosphorylation occurs. You have a look, the joining of a phosphate group to the inactive kinase, and whenever the joining is happen, the active phosphorylase kinase happen, and it in the next step you can see there are stepwise activation 1 2 3 4 5 6 7 8. So stepwise, as for if you just enter a room and you have to switch on all the lights in that room, you have to switch on one by one, 1 2 3 4. Here also 1 2 3 4, all the signaling cascade is stepwise on, and the metabolites produced by the previous signaling step, it enacts as the trigger for the next one. And in the last step, the inactive glycogen phosphorylase, another enzyme, it is triggered, and it is also ATP driven, because in each step for triggering phosphorylation we are just discussing. So phosphorylation means adding of phosphate, and it needs some energy. ATP gives the energy, and the last one, the active glycogen phosphorylase. So this is the last enzyme which has to enact for glycogen breakdown. Glycogen is the component metabolite of glucose, and it for cleaving the glucose, it is a macromolecule to the micro one, the smaller fragment. The glycogen is broken down, and you can see all the cell signaling cascades, they are receptor and signal molecule driven. And it is the last portion of our discussion that many times final response of a cell comes after integrating multiple signals. It is very less of, yeah, I would, please mute yourself. Please mute yourself. So in the left hand panel A, the plasma membrane receptor, somebody is not muted, please mute. The receptor is recognizing the biomolecule, and the cascade, signaling cascade, which is,
Ma'am, I think you have to mute himself. Subroto Pal. I think he's unmuted by mistake.
he himself has to mute.
No, ma'am. Since you are the presenter, you can mute him.
কোথায় রে? সুব্রত পাল।
ওই যে সুব্রত পালের ওইখানে তিনখানা ডট আছে, ওখানে গিয়ে আপনি মিউট করতে পারেন।
Mute participant, it is
হ্যাঁ
muted.
মিউট হ্যাঁ
Okay, done. So the many times the final response, it involves multiple signaling cascade on, and all the responses, all the signaling cascades on behavior, it involves the ATP investment and the phosphorylation. When we target to study, you just see each and every phosphate has to be added by putting some energy. And so from ATP to ADP, adenosine triphosphate to adenosine diphosphate is produced, and the signal relayed onward. So it is looking like this, and here double molecular cascade is on, and here two X and Z components, these subcomponents, they just join together, and they join to form a complex molecular complex, signaling molecular complex, and with the signal produced through the molecular complex, it is relayed onward. And whenever all the signals which are needed to be passed to a cell to get triggered to give some response, in the final step the cell response, and the cell response, I am just going to a previous slide to give you the idea how the cell is responding. Likewise, this the cell responses, some cell just release some chemical like sweat, sebum, some cell release vesicular structure of their epithelial portion encapsulating the chemical like milk from the mammary gland, and the holocrine they are just releasing some biochemical molecules, and after releasing, the cell itself dies, like the sebaceous glands. So it is the whole chapter of the cell signaling, and we will be continuing in the next class the immunology module. Today, do you have some time? I think Nona.
Yes, no, ma'am. We have a class, sir.
Okay,
We have a class
okay,
after on auditor, sir.
okay, okay.